advancing in vitro disease modeling with mouse hepatocytes. Primary mouse hepatocytes isolated from mouse livers provide a complementary preclinical model for human hepatocyte in vitro models. Mouse hepatocytes also have reduced lot variability, are more easily accessible compared to human hepatocytes, and are readily amenable to ex vivo genetic modification. This makes them very useful for studies involving gene editing, gene delivery, and gene targeting. Additionally, mouse hepatocytes provide a predictive link to in vivo mouse models, improving the translational relevance and impact of preclinical research. Ex vivo genetic modification of primary mouse hepatocytes is a robust tool for disease modeling and therapeutic target validation through gain and loss of function studies. This model also supports the development and evaluation of emerging gene therapies, including antisense oligonucleotides and siRNAs. Primary hepatocytes can be isolated from various mouse strains, with C57BL6 and CD1 among the most commonly used for studies involving liver metabolism, toxicity, and gene modulation. Each strain exhibits distinct genetic backgrounds, immune responses, metabolic profiles, and disease susceptibilities, factors that can significantly influence hepatocyte function in vitro. In gene editing application, C57BL6 is often favored due to its broad compatibility with CRISPR technologies. However, hepatocyte isolation from this strain poses greater technical challenges, typically yielding lower cell numbers and viability. These cells also demonstrate reduced plateability and are more fragile in culture compared to the more robust outbred CD1 strain. Beyond strain-specific differences, species-specific factors also present unique challenges when working with primary mouse hepatocytes. Compared to hepatocytes from larger species, such as human or non-human primates, their high metabolic rate and oxygen demand makes them more difficult to maintain in culture. Moreover, mouse hepatocyte cultures often exhibit poor plateability, along with reduced viability and functionality over time, which poses challenges for in vitro applications and longer-term studies. Therefore, optimized isolation protocols are critical to ensure high-quality mouse hepatocyte preparations with robust plateability, viability, and functionality, especially for gene targeting and other downstream applications. To overcome the inherent challenges associated with mouse hepatocytes, we have optimized our isolation and cryopreservation protocols to consistently achieve high cell viability and yield. In addition, culture conditions, including specialized media and plate coating matrices, have been carefully refined to support improved plateability. When cultured successfully, healthy mouse hepatocytes, regardless of strain, display a characteristic hexagonal morphology with well-defined cell borders, indicating proper attachment and viability. Adapting the primary mouse hepatocyte model to a high-throughput platform offers significant advantages for researchers developing next-generation gene therapies. We have established an optimized, high-throughput workflow compatible with automated liquid handling systems for lipid-based transfection of primary mouse hepatocytes to support gene targeting studies. In this automated workflow, hepatocytes are seeded into multi-well plates, and test compounds are introduced via transfection. After incubation, total mRNA is extracted and reverse transcribed into complementary DNA or cDNA. The cDNA is then used as a template for quantitative PCR to assess target gene expression and evaluate the efficiency of gene modification. Here we show mouse hepatocytes from both CD1 and C57BL6 strains, imaged prior to treatment and 48 hours post-transfection using our gene editing workflow. As shown, the hepatocytes remain well attached and maintain healthy morphology following treatment, indicating that our transfection protocol is well tolerated by mouse hepatocytes, even after extended incubation. To demonstrate the effectiveness of our optimized transfection workflow, we assessed gene knockdown of two commonly used RNAi controls, GAPDH and MALAT1, in primary hepatocytes from both CD1 and C57BL6 mice. At the top, GAPDH, a highly expressed housekeeping gene, shows strong mRNA knockdown at both 1 and 10 nanomolar siRNA concentrations across both strains. At the bottom, we see similarly effective knockdown of MALAT1, a long non-coding RNA often used to evaluate RNAi performance. These results confirm that our high-throughput transfection workflow delivers robust and reproducible gene silencing in primary mouse hepatocytes, supporting their use in gene therapy research. As we've discussed, primary mouse hepatocytes are valuable in vitro models, but overcoming their inherent challenges is essential for reliable results. Using high-quality hepatocytes with strong viability, platability, and functionality is critical to experimental success. Optimized culture and transfection protocols help ensure consistent gene delivery, maximize cell health, and maintain data integrity. Incorporating automation further enables higher throughput workflows, shortening timelines and expanding experimental possibilities. Together, access to robust hepatocyte models and streamlined workflows empowers researchers to advance from hypothesis to insight more efficiently and confidently. Thank you for watching. Discovery scientists continue to innovate and deliver solutions that address your research needs. If you have any questions, please email us at info@dls.com.